Hello everyone, our team made up of Carlos Alberto, Ana Paola, Jacqueline, and Eduardo. We'll tell you about food-grade pigments from streptomyces isolated from the marine sponge, Calispongia diffusion. Next, introduction. Microorganisms produce food-grade pigments such as carotenoids, melanins, flavins, quinones, and more specifically, monachids, violacine, indigo, etc. The production of food-grade pigments by microbial fermentation is an developmental stage. Colorants become the most sensitive part of any product, not only because of their attractiveness, but also because of their acceptability by the consumer. For the biotechnological production of such colorants, microorganisms are more suitable due to factors such as ease of availability and cultivation, the potential of microbes to be genetically manipulated, etc. Objective. In this article, we describe the production of carotenoids from Streptomyces strain of white series isolated from marine sponge by fermentation under fluorescent white light.
Materials and methods. Insulation of streptomyces associated with marine sponges. Marine sponges were collected at a deep of 5 to 10 millimeters of Kovalam coast, situated on the west coast of Kerala from India. The sponge extract was obtained by squeezing gently with a glass stick. A liquid's 1 milliliter of aged sponge extract were serially diluted, mixed and with 15 milliliters of glycerol asparagine agar medium and powder into petri dishes and incubated at room temperature of 18 plus minus 2 degrees Celsius for 7 days. Next, please. Um, the identification of a sponge. The sponge tissues were preserved in 10% formalin and photographed by the camera lucida microscope. The spicules were separated from the sponge and fixation was carried out. Spicules or sclerites are skeletal units that are part of the poriferous. The characterization of the streptomyces isolated. The streptomyces was characterized according to the methods employed by collaborators in International Streptomyces Project. The microorganisms was characterized by acid-fast staining and by gram staining techniques. Next, please. The characterization of the streptomyces isolate. The isolate was also studied by employing various parameters, which are influence of carbon source, amino acid influence, sodium chloride tolerance, melanoid production, and the pigmentation of mycelia and spore morphology.
Or the fermentative production of carotenoid pigments. The Streptomyces isolate was inoculated in 500 millimeters Allen-Meyer flax contained 100 of Bennett sugar medium with a pH for 7.2 and incubated at 180 RPM at 28 centigrades for 120 hours in a rotatory shaker with fluorescent white light exposure. Um, the fermentative production of carotenoid pigments. The carotenoid fraction was started by the method described by Schulman. Schulman. The fermented broth in the flask was lympholysed and that extracted with methanol contained 6% of KOH for 15 minutes at 60 centigrade. The carotenoids were protonated using 10% of petroleum. Either the upper phase was colored, evaporated to brightness, and the ratio dissolving in acetone. The concentrated preparation was subsequently purified on silica hot column and eluted with exent toluene. In order to obtain the final carotenoid fraction, a qualitative analysis was performed by silica gel thin layer chromatography with a solvent mixture, and the separation of carotenoids was carried out by HPLC or high performance liquid chromatography.
The marine sponge collected for the insulation of associated streptomyces was identified as Calispongia diffusa, belonging to the class Demospongia solas. Each species have unique species and according to that classification of species can be done. Some of the characteristics of streptomyces installed from marine sponge are shown in the next table. Next, please. At a sodium chloride concentration of 2.5%, profuse runoff of the insulate was observed. The dry weight of the biomass at this concentration was 0.102 per gram. It has been reported that some sodium chloride sensitive actinomycetes have increased tolerance to higher concentration during successive cultivation, and the field rate of the tolerance strain had a significantly different absorbance in the visible spectrum, suggesting the production of new metabolites. Next, please. In streptomyces, carotenoid production is a widespread metabolic activity which occurs in a constituted light-dependent or cryptic manner. Production of the pigments was found to be initiated after 4 days of growth under illuminated conditions. The wavelengths at which the peaks formed were 445 and 471. These wavelets indicate the presence of lycopene and a unidentified pigment. Next, please.
In conclusion, the number of approved colorants for food industry is limited. Some approved food colorants are known by their chemical name, while others are known by her soul. The biocolorants identified by their chemical name can be synthesized easy by cheaper biotechnology, so the sponge-associated streptomyces insulate, uh, which influence like explosion exhibit carotenoid production. This is also reference. Thanks for your attention and see you in the next time.